Hi, this is GCSE 9 to 1 Crammers. It's never too late to start revising, and this way you can do it anywhere. Listen to these questions as many times as you need until the answers pop into your head. Today we're revising biology, topic 2C, enzymes, covering enzymes and what they are, the lock and key mechanism, and optimum conditions for enzymes to work in. What are enzymes? Enzymes are large proteins, biological catalysts, which speed up reactions without being used up. Give 3 examples of digestive enzymes that help to break down different food types. Amylase, which breaks down starch into sugars like glucose and maltose. Protease, which breaks down proteins into amino acids. And lipase, which breaks down lipids or fats into glycerol and fatty acids. What is a catalyst? It is a substance which increases the speed of a reaction without being changed or used up in the reaction. What is the substance that an enzyme acts on or metabolises called? The substrate. Give 3 examples of substrates that enzymes break down in the body: starch, proteins, and lipids. Describe the shape of an enzyme. An enzyme has a lock and key. The lock is the active site, which has a unique shape that will only fit onto the key, which is the substrate involved in a specific reaction. Explain why enzymes only catalyse with one specific reaction. This is because for the enzyme to work, the substrate must fit the active site. What happens to the substrate after it is catalysed in a chemical reaction? The substrate is converted into a specific product: sugars, amino acids, or fatty acids. What happens if the substrate doesn't match the enzyme's active site? Then the reaction won't be catalysed. Therefore, no reaction will take place. What does catalyse mean? To catalyse means to cause or speed up a reaction by acting as a catalyst. What happens to an enzyme after it is catalysed in a chemical reaction? Enzymes are not used up in a chemical reaction. They remain unchanged and can be reused. What is the name of the part of an enzyme that binds to the substrate? The active site. Describe what enzymes are and explain how they break down molecules using the lock and key mechanism. An enzyme is a biological catalyst, so it speeds up a reaction without being used up. They connect to the substrate and break down big molecules into smaller soluble ones. This is the lock and key theory. After the product is made, it breaks away from the enzyme, ready for another substrate to break down. The optimum conditions for enzymes. Name the 2 optimum conditions that are best for enzymes to work in: temperature and pH. What temperature are enzymes most active? Their optimum temperature. It is at 37 degrees centigrade. What happens to the enzyme if the temperature increases above its optimum temperature? The enzymes are denatured. Explain what happens to an enzyme if it is said to be denatured. Some of the bonds holding the enzyme break. This changes the shape of the active site, so the substrate won't fit anymore. Explain how temperature affects enzyme action using this graph. At first, as the temperature increases, the rate of reaction increases. This is the enzyme's optimum temperature, where the enzyme works best. As the temperature keeps increasing up to 40 degrees Celsius, the rate of reaction decreases because the enzymes are denatured. What is the optimum pH most enzymes work best at? pH 7. Do all enzymes work best at pH 7? No. Explain why not all enzymes work best at pH 7. Well, it depends where in the body they work. For example, enzymes in the stomach prefer acidic conditions, around pH 2. Name the substance that is produced to give the correct pH for enzyme action in the human stomach. It's hydrochloric acid. Increasing the temperature increases the rate of reaction. Explain why. Because the reactants have more energy, so they move about and collide more often with each other. Explain how a pH which is too high or too low affects enzyme action using this graph. If the pH is too high or too low, the rate of reaction will decrease and the enzymes are denatured. The bonds holding the enzymes together break, changing the shape of their active site so the substrate doesn't fit anymore. What is the temperature or pH at which an enzyme works and is most active known as? It's optimum temperature or optimum pH. A low pH means that an environment is too acidic. A high pH means that an environment is too alkaline. In an investigation, a glass tube containing boiled egg white was put into a test tube. A solution containing pepsin at pH 7 was added. 6 more tubes were set up with solutions of pepsin at different pH values. The test tubes were left for 24 hours at room temperature. The table below shows the results. At which pH did the pepsin work best? Visualize the results from the table on a pH graph. The starting length of the egg white is 50 mm. At first the rate of reaction increases, with the length of the egg white shrinking to 38 mm, and then to 20 mm in length, before the egg white starts expanding to 34 mm in length, showing the reaction is starting to slow down until it finally reaches 50 mm in length again. Looking at the graph, the optimum pH for pepsin to work at would be at 20 mm, and looking at the table, that is pH 2. Explain why there was no change in the length of the egg white from pH 5 to pH 7. Looking at the pH graph, pH 5 to pH 7 is at the end of the reaction, so the pepsin enzyme denatured, so it no longer fits the substrate because the pH went too high. In this video you've learnt about the enzymes, the lock and key mechanism, about the active site and substrate and the optimum conditions for enzymes to work in, and how enzymes can become denatured. Take time to learn this topic so you can nail those questions in the Biology GCSE Paper 1 exam. The key to exam success is learning, consolidation and keeping this topic fresh in your mind. How do you do that? Repetition, repetition and repetition. If you go over this topic enough times, you'll be able to answer any questions on this topic confidently in your exam. This is GCSE 9-1 Crammers, and if you like the content of this video, give us a thumbs up. And if you have a question or need help, give us a comment below. And don't forget, smash that subscribe button if you want more Crammers videos for your exam success. Thanks for watching and see you again soon.